Good evening, everybody. I hope I'm audible to all of you. We will wait for some time and let everybody join, and then we will begin with today's session. It is very similar to whatever we had earlier. We will discuss a little bit of a brief summary of what is there on this week, and then we will discuss the questions and the answers basically. So it's an interactive session. I would expect all of you to interact in every way possible uh, during this entire session. Okay, good evening, everybody. Today is week 4 of the live interactive session of the course Genome Editing and Engineering, which is taken by Professor Utpal Bora of IIT Guwahati. I am Sanket Mukherjee from School of Biological Sciences, IIT Delhi. So today is week 4 that we will be discussing, and you have already covered 1/3 of the course. So I hope you're enjoying this course very much, and we will see what we have to discuss in this week. So there are only two lectures essentially which talk about targeted gene modification. When we talk about targeted gene modification, we are talking about modifying genes through subtle mutations. Um, there could be insertions, deletions, inversions, all these kind of things which can be introduced. So even like before in the previous weeks, we have already discussed what are the tools that could be used for doing these processes. What we will see today is how specifically these processes are carried out. So when we are talking about targeted gene modification, it involves insertion or deletion of protein coding sequence at a particular locus of an organism's chromosome, right? Um, Usually, whenever we are referring to a gene, as we have already discussed this, I think in the first week, that it usually refers to a protein coding element, right? So when we are inserting something or deleting some part of it, that's what we mean by modification. Modification can be carried out in either in germline cells or the embryonic stem cells. And we will discuss how both of these are, how they are important, how they are different. But ideally, the objective is that wherever you do this modification, it will be transferred from the parent to the offspring. So that is ideally the intention of doing any kind of modification, gene modification. Now, when we are talking about germline gene modifications, we are referring to germline cells, which are either sperms or ovum. These two cells, egg cell or the sperm cell, which is modified, and later fusion of these two cells leads to a new organism, and we get the modified gene in the new organism. This is routinely practiced in case of multiple biological experiments which are which involve biological animals like mice models, if we are talking about them, or hamster models, whatever is used, we prefer germline editing, one of the strategies. Another strategy is involves embryonic stem cells. So stem cells are essentially pluripotent cells. And what do we mean by pluripotent cells? They can differentiate into multiple lineages, right? So in usually an adult cell has a limited functionality, limited structure. So let's say a cell like a skin cell has the same DNA, has the same genetic material to that of a liver cell. However, a skin cell behaves very differently from that of a liver cell because there is some process which we call as differentiation. Now, this differentiation leads to directed functionality of a particular cell. But these cells, they all come from a single parent cell, and that we call as the stem cell. So an embryonic stem cell is usually found in the blastocyst stage, in the inner cell mass of a blastocyst. So if this is the blastocyst, which forms from the zygote, the inner cell mass has the embryonic stem cells. And we take these cells and we replace these cells with the modified cells. This is another very important strategy for gene modifications. And although genetic modification as such is not approved in therapy, there is nothing as such, but still there are multiple drugs, I would say, multiple gene therapy trials which are going on, which are undergoing, in a hope that we will someday be able to cure a lot of genetic diseases based on this therapy, right? Very important things that we need to know about gene modification is we have two objectives, either gene knockout or knock-in. I've already discussed about it earlier also. Knockout refers to deletion of a particular gene. If a gene is not present in the system, what would be its effect? That is how we remove that gene through excision, and we would see what is the effect. Similarly, a gene knock-in would refer to insertion of a particular gene which is ordinarily not present in that particular patient or organism, and we would have a knock-in of that gene. So that's how it is. Now, coming to the next slide, uh, about gene knockout. So, sorry, it's not visible again. Yeah, in this slide, you can see that in a wild type mouse, let's say there is a gene of interest which is functional, which transcribes, which translates, and it has some particular role in its it's based on your biological experiment, right? But when you do a knockout of this gene, let's say this gene is X. So this, if gene is knocked out, this particular gene is no longer expressed. This particular gene, there is no transcription, translation. As a matter of fact, if it encodes some protein, that protein will not be encoded any further. So this is ideally what a gene knockout looks like. Similarly, in case of gene knock-in, what happens? Let's say there is a gene of interest which is present in the mouse. I'm not sure if you can see. Yeah, if there is a gene of interest which is present in the wild type mouse, it is not changed at all. And you would introduce another gene, let's say a gene Y, in this place of in this gene, and that would be gene knock-in. So you are introducing a foreign gene which is not ideally present, and probably you would see what is the function of that gene in further experiments. So this is the basic idea of knock-in and knockout. Now, if we talk about applications, so as we already have mentioned, that gene modification in germlines of mammals, that is one of the modification. Loss of function mutation in knockout while gain of function in knock-in. This is ideally how experiments are carried out. If you want to see whether a particular, let's say, if we talk about cancer, oncogenes, they promote cancer, tumor suppressor genes, they reduce they suppress cancer. So if we want to see the loss of function of a tumor suppressor gene, we would knock out this gene and we would probably see a more progressive form of cancer, a cancer which is more serious. Similarly, an oncogene, if it is not ideally present in the system, if it is introduced, let's say in mice, we would potentially see an accelerated form of cancer. So these are some of the experimental ways we use knock-in, knockout. Function of specific gene in the organism could be figured out. So what happens when there are multiple we have like in humans there are so many genes that are which encode for particular protein, but we for all genes, I mean how to find out that what is the role of this gene, whether in case of development, whether in case of any other disease progression or any other case homeostasis. So to study the function of these of any specific gene, we can knock out that gene or knock in that gene to see what is the role of that gene in that organism. Transgenic animals are created using gene knockout or knock-ins. Disease models are created. So now, if you might have heard about NOD/SCID mice, which are routinely used. These are mice models which are immunodeficient. So these kind of mice models are created using gene knockout or knock-ins, right? Several medical therapies could be a potential application of gene knock-in knockout. So essentially, we can reverse a mutation, or for example, in case of sickle cell anemia, where there is a point mutation, you must be aware of it. Similarly, in case of beta thalassemia, so sorry. There is a mutation which is present. So, so to reverse those mutations, it is possible to reverse those mutations if we use gene therapy. However, it is still not in practice. So in 2007, Nobel Prize in Physiology and Medicine was awarded to three scientists, Mario R. Capecci, Martin J. Evans, and Oliver Smithies, for the discoveries of principles for introducing specific gene modification in mice by the use of embryonic stem cells. So they were the first group of scientists who were able to modify a mice. So we call these kind of organisms as genetically modified organisms, right? And they use the embryonic stem cells technology. So this had led to the creation of an immensely powerful technology referred to as gene targeting. It is now widely used. It is now widely used um, in understanding the function of a gene in health and diseases, these all things. So till date, we use genetically modified organisms, specifically mice models, which are very close to humans, for biological experiments. Right, we will see the contribution of each of these scientists. So, Oliver Smithies, he established that homologous recombination, it causes allelic variation in human haptoglobin genes. Right, so we have already talked about homologous recombination, how the process works. We already know that. So essentially, if there is a damage is in one of the DNA strands, or one of the alleles of the DNA, we would say, of a chromosome, it will essentially have another one as a template, and there will be homologous recombination. So another template which is undamaged will serve as a template for recombination, and this is basically a damage repair pathway. So through damage repair, through this mechanism, there are sometimes introduction of some mutations, or I would rather say variations. So these variations are introduced in the new DNA that is formed, which is the repaired form or version of DNA. And these variations are essentially very important if you have studied some basic biology, that variations are very important for survival. It brings about a lot of changes in an organism, which is important for it to adapt to its environment, all these things. So similarly, Wigler and Richard A. Axe in 1970 did a phenomenal job of regaining enzyme thymidine kinase gene in mammalian cultured cell. He introduced functional copies of kinase from herpes simplex virus. Herpes viruses are a group of viruses which are abundantly found in humans. They usually infect. They usually infect every human. So these from this virus, they got the functional copies of thymidine kinase gene, and they introduced these genes to a cell line that did not have this gene. So this gene was taken by phagocytic uptake. However, the efficiency was very low. Very few cells were able to take up this gene. Nowadays, when we perform gene insertion experiments, usually, if you remember the plasmids we had already discussed in one of these slides earlier. So plasmids are usually inserted. If you remember, there are shuttle vectors which can work on more than one organism. So these kind of plasmids, which could be shuttle vectors as well, they are usually inserted into a cell line or even in a bacterial cell as well. So we could do that. There are various processes like transformation or transfection, which is used. So but the idea is same, that a gene that is not present in the cell is introduced from a foreign source. That is the basic concept. Now, Mario Capecci, he solved the problem of this low efficiency of efficiency by devising a method by introducing DNA directly into the nucleus by bypassing the cytoplasm and lysosome through injection by a tiny glass pipette. So we also call this as microinjection, and it's still a practice in some of the labs. It's still preferred. So which kind of mechanism you would use to insert a gene, it greatly depends upon a lot of factors. But microinjection was one of the things that he discovered. Now, previously, the efficiency was like 1 in million cells. Now he was able to increase this efficiency to 1 in every 3 cells. So that was pretty good. But anyway, it's anyway a low efficiency. But whenever you try to introduce anything foreign to a cell, it's more than likely that the efficiency would be low, right? That's how we see in normal experiments as well. Martin Evans, so we will see his contribution. So Capecci and Smithies carried out genetic modifications in somatic cell lines. The revolutionary work of Martin Evans on embryonal carcinoma cell lines enabled their work to be carried out in germline cells. So even Evans' work on embryonal carcinoma cell lines derived from mouse testicular teratocarcinomas. These cells can be induced to differentiate into multiple tissue type. Through cell culture, Evans characterized the teratoma-derived cells of mice strain 129 SV that have a high frequency of tumor harboring totipotent cell. Now, you would ask why, uh, you know, uh, doing any kind of genetic modification in a somatic cell doesn't make any sense. So if you remember, I mean, if you know the difference between a somatic cell and a germline cell or an stem cell, so what are these cells? What is the differentiation between these cells? So somatic cells are all those cells which are not involved in any process of reproduction, right? That is these are basically somatic cells. These are normal cells which are present in your skin, your liver, all the other organs. These all cells are somatic cells. Now the problem with modifying somatic cells is that uh, they have, I would say, limited replicative ability. So let's say, for example, if you are talking about a disease, a blood cancer, okay, so that has been caused due to some sort of mutation in some particular gene. So let's say there is blood cancer. Now, if you are going to modify an adult, or I would not say, let's say, if not about blood, let's say about skin cancer, any form of skin cancer. So if you're trying to modify individual skin cells, it's a very hectic process. It's not possible. But when we go for modifying a stem cell, there it is possible to create some impact, because that stem cell will further divide into multiple cell types. So there are like adult stem cells also. So there are modifications in adult stem cells which could be performed. So let's say every cell type has its own adult stem cell. If we modify this adult stem cell, there is a chance that it will further differentiate into its own lineage, and it will lead to multiple cells which would have this corrected version of gene. However, if you uh, edit an adult cell, it will only be effective for a few cycles of replication, and basically the your corrected gene will not spread out in the body. That's ideally that's the idea. Talking about germline cells, these are cells which are present in the testes and the ovaries of humans, and even in case of plants, these are basically the sex cells which we would call. So ideally the egg and the sperm have these cells. So when you modify germline cells, there is a higher chance that the new organism or the new offspring that you get would have the corrected version of the gene. Stem cells, on the other hand, if it's routinely practiced, that like we talked about embryonic stem cells. So embryonic stem cells, if they are modified, which is essentially when your cell, when the zygote is in its blastocyst stage, when multiple differentiation has not happened, if you make any modification at that stage, it is very likely that when those cells will divide, they will lead to creation of the cells which have this modification, your intended modification, in the offspring. But there are some ethical concerns which are related to it. That's why there are, iPSCs, that is induced pluripotent stem cells. So essentially, an adult cell can be converted to an pluripotent stem cell through introduction of certain factors. So that also, I think, we will touch upon in later lectures a little bit, but that is also one of the methods that can be preferred to introduce any modification, any genetic modification in the cells. So anyway, if you look at this diagram, then you would see that Martin Evans, what he did, he took this is the basically fertilization event where oocyte and a sperm, they fuse together to form a zygote, right? Then the blastocyst is formed, one of these stages in a human fetus, and from there the inner cell mass is taken, which is pluripotent. Fine. So these pluripotent cells can lead to generation of multiple cell types. As you can see, the circulatory system, nervous system, immune system, it could lead to multiple cell types. So if you have a very specific gene that you want to introduce at a very early stage of life, in a hope that all the cell types, all the cell types will have this gene, then it is ideally preferred to be done at this stage, at at the blastocyst stage from the inner cell mass. Okay, another thing is, what is the difference between totipotent and pluripotent? So essentially, when we are referring to any stem cell, we mention the word pluripotent because it can form three germ layers. Essentially, the ectoderm, endoderm, and mesoderm can be formed using from a pluripotent cell. What a totipotent cell is, it is a pluripotent cell. So it's basically a pluripotent cell, plus it can also form placenta. So usually totipotent cells at that stage, we do not disturb the fetus. It is because these processes are disruptive processes. So a placenta is usually only required during the birth of an organism, whether we are talking about mammals here. So only at that time a placenta is required. Later in life, you would not require a placenta. So you would not like to edit a totipotent cell, but rather a pluripotent cell. So that's one thing. So Evans was able to create chimeric mice. Chimeric organisms means when there are multiple genes in an organism. So multiple cell types, if there are in an organism, those are called chimeras. So he injected cultured embryonal carcinomal cell into mouse blastocysts, which were then implanted into a foster mother. It resulted into development of a chimeric mice line that contained tissues of cultured carcinoma. In another study, when the embryonic cells to be injected were infected with retrovirus, the retroviral DNA was detected in both somatic and germline cells of the chimeric mice. So retrovirus is again a method. So one can use plasmid, one could use, gene insertion into the genomic DNA. One could use retroviruses, which would be integrated into the genomic DNA. So there are multiple ways of doing it. We have already studied about it. This is a one slide which is very similar, which we have already talked about in the previous lecture also. So serine type recombinases, when there is 180-degree rotation uh, of uh, the strands, which causes a recombinated strand to be formed. Similarly, there is tyrosine recombinases, which requires four. Basically, it works in quad system, where there is formation of a holiday intermediate, and then there is a recombinated DNA that we get. So these techniques will be very important to introduce the genetic modifications in a system, in a cell. So for example, tyrosine recombinases, we have already talked about pre-recombinase, FLP recombinase. These are tyrosine recombinases. We will see how these are used. So in case of pre-recombination, so in the first generation, you see, so there is a pre-recombinase gene. So this gene will basically encode the pre-recombinase protein, and your gene of interest is fluxed by the LOXP site. Right, these are the LOXP sites which will be present on the exons, on the either end of the exon. So what is an exon and what is an intron? Exons are part of a gene. So if you remember that in a DNA, let's say if this is a DNA, I'm drawing it as a line. So several parts of it are expressed, the rest are not expressed. So this happens due to the event called splicing. Right, you must be knowing this. This is very basic molecular biology. Now, the ones which are expressed, they are called exons, right, which code for proteins. The ones which do not, they are introns. So essentially what happens from a DNA to an mRNA, this splicing process occurs. Now, when an mRNA is formed, it will only have the exons. right? So it will only have these exons. Introns will be spliced out. So there are various proteins which are involved in splicing as well. When we create DNA out of this mRNA, if we use reverse transcription, this DNA is called a complementary DNA, cDNA, right? So why I explained this? Because in either end of an exon, let's say if you want to remove, this is exon 1, 2, and 3. Now, if you want to remove exon 1 from a gene, you would introduce the LOXP site on either ends of this exon. And when you have this on the either end, we say that this exon is floxed. So this term comes due to the LOXP term. Now, once the Cre recombinase is secreted, then um, this LOXP site will be removed ideally. So it will be excised. Similarly, for FLP, FRT recombinase, also known as flipase. So here, FRT is similar to LOXP in case of Cre-LOXP system. So FRT is again, then it is present on either end of an exon, and FLP is the recombinase enzyme. So whenever a recombinase is produced, it will cause excision of that fragment. So similarly, based on this, Cre-LOXP system, we have the TET on, TET off system, where TET stands for tetracycline. So tetracycline are a class of antibiotics. So whenever in a TET off system, what happens is, whenever this antibiotic, for example, doxycycline is a tetracycline antibiotic. So whenever doxycycline is introduced into the system, then in a TET off system, the gene regulation, basically the gene will stop expressing. As you can see in the figure A, whenever doxycycline is introduced in a system, then transcription does not happen. But when this doxycycline is absent, transcription happens, translation happens. It's a normal gene function. Similarly, in a TET on system, whenever there is induction or introduction of doxycycline, there is induction of transcription. When there is absence of doxycycline, there is no transcription. You can read about these processes in details in the lecture, but this is the idea. Now, how to design a vector for knockout? So as we already know, like a normal vector, which we have already discussed the characteristics of a normal vector, we would have a selectable marker gene. Okay, so essentially this selectable marker gene could be an antibiotic resistance gene, which would let you know that your gene of interest, or let's say knockout or knock-in, has happened or not. How would you know that? For that, you need a selection marker gene. So positive selection genes are like neomycin, puromycin, and hygromycin. Addition of herpes virus thymidine kinase gene adjacent to one of the vectors homologous arms help in determining homologous recombination or random integration. Now, random integrants will usually contain an intact copy of HSV thymidine kinase gene, whereas um, the ones which will have homologous recombination, they will not have this intact copy. Now, cells with random integrants are killed during negative selection through the treatment of gancilovir or FIAU. Presence of thymidine kinase causes phosphorylation of these compounds, which inhibit DNA synthesis and leads to cell death. So similarly, we have replacement vectors. So what do replacement vectors do? So if there is a target or a gene which you want to replace with some other gene, it will cause homologous recombination because it will have some elements which are common in both the cases, and then this gene will replace the target gene. So that is the replacement vector. Similarly, we have insertion vector, which basically integrates DNA into the host genome, right? So, for example, we have talked about lambda phages. Lambda phage is essentially one of the strategies how it causes again recombination and leads to insertion of the phage DNA into the bacteria. Now, subtle mutations, basically mutations are introduced by either replacement or insertion type vectors. So it can introduce desired subtle mutations, which are like point mutation, microdeletion, insertion into the target gene. So there are several processes which could, I mean, several mechanisms through which you could introduce subtle mutation. One of them is hit and run approach. Another one is tag exchange approach. And the third one is recombinase based approach. So we'll talk about all three of them uh, very quickly. Okay. So in a hit and run approach, there is an insertion vector, which is used for, homologous recombination. If you see here, there's an insertion vector, which is used for creating homologous recombination, out of which the gene of interest is now inserted, right? The gene of interest is inserted into the genomic locus. Now, what you want in the next step is, now this gene of interest will also have some selectable markers like neomycin and the thymidine kinase. These are selectable markers. You do not want these selectable markers to be present in the final gene. You want to remove it. So for that, there is the run step where again, there is intrachromosomal recombination, and then we remove the selectable marker through this recombination step, and there is this gene which will have your inserted product, right? Similarly, there is tag and exchange approach. So here there are two types of vectors, vector 1 and vector 2. Right? Two vectors are used. And in the first step, there is homologous recombination with vector 1, so which we call as the tag step. So this is vector 1. There is homologous recombination with vector 1, which leads to the replacement of okay, wait. Yeah, so there is replacement of your of the gene with the gene of interest. Then there is the vector 2, which will have again homologous recombination and site-directed. It will basically consist the gene of interest. So this will again exchange with the previous one, and then we will have the genome swapping. So this is tag and exchange approach. Recombinase approach, we've already talked about Krelok system. We talked about FLP, FRT systems. These are how recombinases can be used for deletions or insertions, as a matter of fact. So for creating a knock-in or knockout mice, how to go about this process. So as we already said, if we are talking about embryonic stem cells, there we are talking about blastocyst, which has an inner cell mass. These are pluripotent cells. We take these cells and then we construct a targeting vector. Now this vector contains your gene of interest, right? This vector contains your gene of interest. Now this vector needs to be inserted into the cell, and this process is called transfection. We have already discussed about it. Then on the fourth step, there is homologous recombination, where the two copies basically. So let's say there is a target gene, and this is the vector. There is homologous recombination where your gene is inserted now. Now then we select these targeted cells through positive and negative selection. So we already talked about positive and negative selection. For example, neomycin resistance, this is positive selection. Thymidine kinase is negative selection. We already talked about it. And from there, we get pure cells which are genetically modified, right? Then we transplant these modified cells back to the blastocyst, and we allow this to grow in a foster mother, and now the new offspring that will be produced through this process will be genetically modified. Okay, let's come to the questions. So I hope everything is clear. If it is clear, you can just give me a like, a thumbs up would work fine. Any questions so far? Okay. So now is the interactive part of the session. So I would expect answers. You could raise your hands and speak, or you could even use the Q&A section for answering. So the first question is, who established that homologous recombination causes allelic variation in human haptoglobin genes? Is it Oliver Smithies, Mario R. Capecci, Martin J. Evans, or Michael Wigler? Anybody would like to answer? No answers? We have already discussed this today itself.
Oliver Smithies.
Yes. Oliver Smithies is the right answer. So, question 2. The inner cell mass localized at one pole of the blastocyst is the source of dash cell lineage: unipotent, multipotent, pluripotent, or totipotent. Answers?
Pluripotent.
Pluripotent. You're absolutely right. Talking about the other forms of the cell types, a unipotent cell is like an adult cell which can only lead to cells of a particular lineage. It is basically a differentiated cell. For example, a skin cell can only divide to form another skin cell. That kind of a cell is a unipotent cell. When we talk about multipotent cell, these are stem cells. These are the examples would be adult stem cell. These are multipotent cell. So for an example of an adult stem cell would be, let's say, neuronal stem cells, or there are even, hematopoietic stem cells. They can lead to multiple cell types, like a hematopoietic stem cell can form RBCs, WBCs, multiple kind of cells, but only the cells that are involved in formation of blood. So that kind of potency is called multipotency. When we talk about pluripotent, we already said it can form all the three germ layers. So ectoderm, mesoderm, and endoderm. It can form any type of cell, basically. It can differentiate into any form of cell. Totipotent cells are basically pluripotent cells, which can also form the placenta. Okay, we have already discussed this. Okay, Cre recombinase belongs to dash class of site-specific recombinases. Is it serine, tyrosine, leucine, or methionine?
Tyrosine.
You are right. Tyrosine is the right answer to this. Cre-LoxP consists of two 13 base pairs dash that flank a central sequence of 8 base pair, which determines the directionality of the LoxP site. Is it palindromic sequence, AT-rich region, GC-rich region, or reverse sequence?
Palindromic sequence.
You're right. Palindromic sequence is the answer. In conditional knockout strategy, the LoxP site can be transferred around an exon of a gene, intron or sequence, 3' end of DNA, or poly A tail. Any answers? No answers? Again, see, this is something we discussed today itself. LOXP sites is transferred around which part of a gene? Exon, intron, 3' end, or poly A tail?
Exon of a gene.
You are right. Exon of a gene would be the answer. Hit and run approach is used to induce deletion, inversion, transcription, or subtle mutations.
Subtle mutations.
You're right. Subtle mutation. Which one of the following option is incorrect regarding the generation of an incomplete gene knockout? Is it alternative or tryptophan promoters, constitutive promoter, differential splicing, or read-through transcription? Any answers?
I'm not sure, but I think differential splicing.
Okay. The question asks which of the following is incorrect in generation of an incomplete knockout. So if there is an incomplete knockouts, let's say if you want to remove a gene X and it's not completely removed, what could be the drawbacks which could happen, right? Let's discuss about it. So there could be a possibility that gene X is expressed but at a very low status because of the incomplete knockout. So because there are uh, expression of cryptic promoters. So the sum of the promoter, it remains in so essentially the entire gene is not removed. Okay, that could be one of the possibilities. Another thing is differential splicing. So what could happen is, let's say a gene could have multiple exons, right? Let's say 1, 2, 3. In the process of creating a knockout, you might have uh, uh, in an incomplete knockout, you might have a differential splicing pattern where let's say 1, 2 remains, 3 is removed, right? 3 is removed, 1 and 2 remains. So that 1 and 2 can also carry out the function, but one of the element is removed. That could happen. Read through transcription is where drug resistance gene. Uh, so yeah, while you are creating a knockout, what could happen? There are like start codons and then stop codon, which start and not codons, start and stop sites of transcription for creating RNAs. During this process of knockout, it is possible that one of these is eliminated, right? Either the start is site is eliminated or the stop site is eliminated. Any of in any of these situations, it might happen that if the stop site is not present, the transcription machinery, it keeps on going because it's not encountering a stop signal. So these three are which are well known things that happen in case of an incomplete knockout. Constitutive promoter, on the other hand, is not an would be the right answer in this case. It's incorrect in this case. So constitutive promoter is the answer. 3 recombinase carries out translocation when LOXP sites are in the same direction, opposite direction, on the complementary strands, or on the same chromosome. Have you read about it? Just for the context, what is translocation? Translocation is, let's say, from one part of a gene, it gets, transferred to some other part of the gene. That is basically translocation. Okay, it could happen in the same chromosome. It could happen in a different chromosome. These kind of events do happen in humans as well. But the question is, when a translocation event happens, the LOXP sites are in the same direction, opposite direction, on complementary strands, or on the same chromosome. They are on the opposite, on the complementary strands. So whenever a LOXP site is in the same direction and on the same chromosome as well, at that time, whenever there is Cre recombinase is introduced, it causes excision, figure A, right? Whenever they are in opposite directions, it causes inversion. When they are on complementary strands, on two different strands, that causes translocation. So this is already present in the lecture. In which phase does the replication of chromosome occur in mitosis? Is it G1, M, G2, or S?
S.
S phase. It's also called the synthesis phase for this reason, because the main material, genetic material, that is DNA, is replicated at this phase. Who created chimeric mice by injecting embryonic carcinoma cells into blastocysts and reimplanting them into foster mice? Was it Martin J. Evans and Mario Capecci, Matthew Kaufman and Mario Capecci, Evans and Kaufman, or Mario Capecci himself? These are about scientists. You can read about them. Evans and Kaufman together did these experiments. Who was the first to use homologous recombination in embryonic stem cell culture to target a mutant hypoxanthine phosphoribosyl transferase gene, HPRT gene? Was it Capecci and Smithies? Was it Smithies, Evans, or Capecci? Any answers? Smithies, Oliver Smithies did this experiment. You can read about it in the lectures. The dash, a 38 kilodalton protein, can perform efficient recombination at LOXP sites in eukaryotic cells. Is it the Cre recombinase, the FLP recombinase, the Cre FLP recombinase, or the LOX recombinase? Any answers? Nobody would like to answer?
Cre recombinase.
Yes, you're right. Cre recombinase. Which of the following statement about the Cre-LoxP system is correct? Cre-LoxP system consists of two 13 base pair palindromic sequence that flank a central sequence of 8 base pair. Cre-LoxP system consists of two 8 base pair palindromic sequence that flank a central sequence of 13 base pair. Production of either excision or inversion is independent of the relative orientation of the two involved LoxP sites, or Cre recombinase is derived from Saccharomyces cerevisiae. Which of the following statement is correct?
Option A.
You are right. Option A. We already discussed this a few questions back. It's just another way of same question. Which of the following is true for floxed mice development for conditional knockout? Phenotypically wild without containing a floxed allele, phenotypically wild with containing a floxed allele, a transgenic mice expressing recombinase, a wild mice expressing recombinase. Which of the following is true for floxed mice development for conditional knockout? Phenotypically wild with containing a floxed allele would be the answer, option B. I'll tell you why. Uh, Whenever, what is conditional knockout? A conditional knockout is when you do not want to knock out a gene uh, right away, straight away. So you do not want that gene to be deleted. Rather, you want to introduce a condition that could be like the tet-on-tet-off system we already discussed. So, for example, introduction of an antibiotic or something else could be a drug, or it could be a pH change that could induce this, deletion of that particular gene. Or it could be stress. So, whenever there is conditional knockout, conditionally knocked out mice, which uses the Kree-Loxp system, it is essentially a phenotypically wild mice. Basically, it's a mice that is similar to wild type. It's exactly the same as wild type. Just the difference is that it has a floxed allele. Floxed allele means in the gene of interest which you're talking about, it will have the flox sites, if LOXP sites basically. The vector for knockout mainly contains two stretches of nucleotides with homology to the dash gene and a selection uh, gene. Basically, what it would be, what will it be? Targeted gene, marker gene, restriction site or marker gene and targeted gene, and marker gene or restriction site.
Targeted gene and marker gene.
You're right. Which of the following is true about homologous recombination through a replacement vector? A negative selection marker should not recombine into the chromosome. The negative selection marker is recombined into the chromosome. This process will result in generation of mutants that promote gene expression from the targeted locus. None of the above. Which of the following is true? I don't know if you can see the question. Yeah.
Option A.
You're right. Option A is the right answer. So the negative selection marker is already, I mean, the reason for a negative selection marker is that these selection markers should not be introduced into, or it should not be integrated into the DNA. If so, these negative selection markers are used to kill the cell, the ones which have integration. Two more questions. Question number 17. Which of the following is true for an insertion type vector for targeted gene modification? The homology arm is absent. It is designed with one arm of homologous sequence. It is designed with both 5' and 3' homology arms, or the homology arm is cleaved by a restriction enzyme.
Option B.
You are right. Designed with just one arm of the homologous sequence. So that's how insertion type vectors are created. Uh, by which of the following approaches subtle mutation can be introduced? Hit and run, tag and exchange, recombinase based approach, or all of the above? Is the last question pretty easy one as such? Any answers? Well, all three approaches can be used, so all of the above is the answer. Okay, that concludes our today's session. I hope you had a good time here. If there are any questions that you would like to ask, now is the time. Any questions? I'll just quickly have the attendance sheet. Where do I get it? Any feedback from your end? Anything? If there is nothing, then I would call this session off, and we will meet again next Tuesday, same time, 7 to 9, and we'll discuss about week 5. So if there is nothing to discuss further, you can leave. Sorry.